All right, welcome back to part 2 of this lesson. Right, so we said the sperm is produced from the testis. So the testis is involved in the process of spermatogenesis. Right, now let's look at how the sperm is produced in there. So in the process of spermatogenesis, we have a cell that we call a spermatogonium. So this one is a stem cell. It's a cell that doesn't undergo differentiation. These are cells that are used in stem cell research to produce other cells. So this cell undergo mitosis, which is the first cell division involved, to produce another cell that is diploid. Then you can tell that due to mitosis, this cell type diploid produce another cell that is diploid. So you can tell that this one is a stem cell. So this cell is called a primary spermatocyte. It is diploid in nature. The primary spermatocyte will then undergo the first meiotic division, that's meiosis 1, to form 2 secondary spermatocytes. And these ones, they are haploid because meiosis results in a halving effect. So this secondary spermatocyte will undergo meiosis 2 to form haploid, again spermatid. Then the spermatid will undergo differentiation or change and develop into sperm cell. So the sperm cell will have head, acrosome, middle piece, head, acrosome, middle piece. And you can tell that the sperm is now different in terms of the structure. If you compare it with the spermatid, the secondary spermatocyte, the primary spermatocyte, and the spermatogonia. So this one is a haploid sperm that is going to be used during fertilization. Right. And we know that this sperm reached maturity immediately after entering the epididymis. Now, so what is the structure of a matured sperm? A matured sperm has a normal shaped head with a sharp pointing acrosome, a middle piece, as well as a tail. Then the middle piece has mitochondrion. The head has a nucleus with chromatin network that are made up of chromosomes that are haploid. Right, so this is a typical structure of a sperm cell, even though it looks like something scary here. Right, so the head of the sperm is made up of an acrosome and the nucleus. The acrosome is sharp pointing. It contains enzyme. The enzyme in the acrosome assists in the penetration of the zona pellucida, which is the outer layer of the egg cell. So this is the structure. The enzymes in the acrosome assist in penetration. And the specific name of the enzyme that is found in the acrosome is enzyme hyaluronidase. Then we have the head, that is the nucleus. We know that in the nucleus carries genetic information. In other words, the nucleus carries paternal chromosomes or paternal genes. Then the middle piece, this one has mitochondrion, and the mitochondrion supplies energy for the movement of sperm. And last, we have the tail. Some they label it as a flagellum. The tail facilitates movement, or it propels the fast forward movement of the sperm. And we know that this is a structure that is formed from the process of spermatogenesis, which is the process I just mentioned, and the sperm will reach maturity immediately after entering the epididymis. Now, sperm enters here, it is ejaculated. Then we have this large gland. This gland is known as a seminal vesicle or a vascular gland. So it has two names. The examiner will decide which one they want to use. So the seminal vesicle, this one is the largest sex gland. So if the question asks you the largest sex gland that resemble the shape of clustered grapes, then you should know that that's a seminal vesicle. So the seminal vesicle, this one, produce a fluid that will provide energy and nutrients. So this one will produce fluid that provides sperm with nutrients. So now you understand that sperm need nutrients, sperm need energy. And we said within the testes, the sperm got its nutrients from the Sertoli cells. Now, since the sperm is outside the testes, it will get nutrients from the fluid produced by this accessory gland, which is the seminal vesicle. Then it will move again. We have this one, the second gland, the second accessory gland, that forms a ring around the urethra. This one is called a prostate gland. So a prostate gland, this one is a gland that produces a fluid that gives semen a characteristic smell. So the smell or the unique smell of the semen is determined by the fluid produced by the corpus gland. Then the last gland is this one that is very close to the anus. This one is known as a bulbo-urethral gland, commonly known as a corpus gland. Right, so this one, the main function, this gland will produce a fluid that will help in what? That will lubricate the urethra. So seminal vesicle produce fluid that will provide nutrients and energy for the sperm. Prostate gland will produce fluid that will give a semen a characteristic smell. Corpus gland or bulbo-urethral gland, this one will produce a fluid that will lubricate the urethra. So collectively, the fluid produced by these three accessory glands, A, B, and C, collectively, this fluid, they will combine and form what we call what? Semen. What is the semen? Semen is the fluid produced by the accessory gland. So the combination of the fluid produced by the accessory glands forms what? Semen. And semen contain what? Sperm. So there's a very huge difference between a sperm cell and a semen. Sperm cell, this one is a male gamete. Semen is a fluid produced by the accessory glands that contains sperm. Right. Then, after getting all the nutrients, then this sperm will enter the urethra. And do not confuse urethra with ureter. So a urethra is a common opening for both urine as well as what? Semen. So if you look at this structure, this one is a structure that is not directly involved in reproduction. It is called a bladder. So it's the one that stores urine. So if you check it, it is in contact with the urethra. So this one allow urine when nature calls, and the urine will bypass in the urethra, and the semen from the sperm duct will also use the same package. Then you can tell that there's no way you can produce both the urine and the semen simultaneously, because there's only one sphincter that controls the exit of both. Right, so this one is our urethra. Then we have this S-shaped structure. You see, the S-shaped structure. So this one is what we call a sigmoid flexure. So the sigmoid flexure is the one that actually gives us an indication. This one is what gives us an indication if the animal is erect or non-erect. So since this sigmoid flexure of ours represent an S shape, so that simply show that the animal is non-erect. Then the erection is controlled by this muscle. It is called an erector muscle. When this muscle contracts, the S-shaped sigmoid flexure change to a straight shape. So this S shape will change and be straight. And if it becomes straight, then we know that the penis will protrude out of the prepuce. So we do not say foreskin. So if this muscle contracts, the sigmoid flexure will change its S shape to a straight shape, and this will cause the penis to protrude out of the prepuce. This shows that the animal is erect. But if the erector muscle relaxes, then it retains its normal shape, its S shape, then the penis will return back there. Then last is the penis. This one is the male organ of copulation. This is the part that is used to deposit semen into the body of the female animal. Danke.